Evening, everybody. Let me know if you can hear me. This should be fine, but just in case. Uh, can you hear me?
Yeah, we can hear you.
Yep. Okay. Alrighty. We'll get started today with I guess the last lecture before the second midterm, which is, if I'm not mistaken, next week. Week 9, right? Um, so same day and time, I mean, as before. We got the rooms booked up. Um, so it's going to be 7:30. Let's just give me a second. I'm trying to. Yeah, I think it is the same day and time. Yeah, 7:30 because for one of my courses, so we start 1 hour earlier for the tests. So 7:30 in room 203, and then that second other room, whatever that number is, same place, same location. The lectures covered will be, it's on Friday, obviously, 7:30 p.m. on Friday next week, week 9. Let me actually um, double check. I'll stop sharing for a second. I'm always, 'cause sometimes we change things and you forget, and then I don't want to announce the wrong date, but I'm pretty sure it's week 9. So, but yeah, it's gonna be same format as last time, obviously. Um, Lectures covered will be pretty much everything we did after the first midterm. So that would be environmental monitoring. That will be a water lecture, and it will be whatever we covered today. Only half of disinfectants lecture. We're not going to be able to cover the whole thing. We never do. It's half of disinfectants lecture for the second midterm always. So 2.5 lectures, right? Checking the course outline. Yes, week 9. Week 9. So everything in order. So 2.5 lectures. Again, I mean, um, not that much at all. I know, people think, well, it's about the complexity maybe of the questions or something, but um, still, it's 2.5 lectures. It's not 5 lectures. It's not 7. Um, in universities, you get a lot more for midterms than 2.5 lectures. Plus calculations, but calculations are going to be exactly the same type of calculations as last time. There were no new calculation type of questions covered or anything like that. So it's going to be exactly the same thing, just different values, obviously. And there will be literally the same type of questions, the same type of problems for the calculation questions as last time, some of them, with like literally just different values, like different numbers. And you have to do pretty much the same calculations, just plug in different values. So as long as you paid attention, as long as you, I guess, um, as long as I guess you knew what kind of mistakes you made, that's what you can come and take a look at the mistakes for. You should be able to do those. And then same idea, it's 2 points for each calculation question and 1 point for each theoretical question. There will be, I think, a little bit more, like 5 more questions in total, something like that. You can see that, you can check that in your grade book if you look at the total marks allocated for the test. Basically, if you look at the overall number, subtract 20, because that's going to be 10 calculation questions, 2 marks each. Whatever number is remaining is the number of theoretical questions, because each theoretical is 1 point. Calculation 10 of them, multiply by 2, gives you 20. Um, the rest of the calculations, so pretty straightforward. Um, Same idea, bring the pencil, obviously bring the calculator. Um, I don't know. I guess I'll post today, tomorrow, the announcement, just in case the same sections that wrote the test in the room 203 will be writing it in the same room 203 next week. The three sections, I think it was, that wrote it in test in the room next door will be writing it next door again. So it's the same number of people, obviously, so the same logistics. Okay. I'll post the answers to the second set of practice calculation problems. Hopefully everybody tried them by now themselves. If you haven't, you can still do it. There's still a week left. Again, can't stress this enough. I've been saying it last time. A very common mistake students are making. They peek into the solutions right away and they get a false impression that they actually understand the calculations. I mean, it's a very common thing amongst students. Try solving everything on your own without peeking into anything at all. That will give you an honest, opinion about whether you understand these calculations or you don't. Because obviously at the exam, there will be no peeking, and then factor in, I guess, the anxiety, the exam anxiety, the fact that you're like limited on time and stuff. So try all those calculation problems if you haven't done so yet on your own before looking into the solutions. If you have any questions, a couple of people did before the last midterm. You can always email me about the calculations, but the solutions pretty much should resolve everything. Okay. So that's that. That's that. So yeah, let's get started with today's lecture. We will, like I said, cover only half of it. About 14 slides, not complicated content at all. And I will also, I think we will have time for it. I will explain briefly the Fresnel coefficient, which is a part of the lecture. The Fresnel coefficient technique, it's one of the lab technique that is used to estimate the efficiency of disinfectants. So it is part of the lecture, and it will be the focus of your next lab next week. So I will explain that today, and then it's going to be much easier. It's not a complicated lab at all. It's a 1 week lab. It's just sometimes people don't get, sometimes I get complaints from the students they don't get the proper explanation of it in the lab. So considering it's a part of the lecture anyway, I try to explain it ahead of time in actual lecture, and then people get also additional explanation in the lab. Alrighty, so disinfectants. We'll talk about disinfectants, preservatives, all sorts of antimicrobials. First part of the lecture that will go towards the second midterm will cover just some general aspects of disinfection, and then the second part of the lecture that will not go towards the second midterm will go towards the final exam. We will look at specific representatives, specific classes of certain antimicrobials, by no means all of them, just selected few, and their mechanisms of action. So that is towards the final exam. The first part is going to be just general information. So disinfectants, right? What are they? We will look at a bunch of, well, I guess 3 major classes of antimicrobials: disinfectants, antiseptics, and preservatives. I'm just showing you all 3. We will concentrate mostly on disinfectants. And then there will be a 4th class, which we will study like in the upcoming weeks, which are the antibiotics, also antimicrobials. So make sure you understand the difference between the 4. Sometimes there is some misconception about which one is which. People, not often, but it does happen. People use these sometimes certain terms interchangeably. So I briefly introduce it here. Disinfectants are the chemicals that remove microorganisms from inanimate surfaces. That's a key word here, inanimate surface. So the non-living surface, so to say, like a bench, a table, a wall, a ceiling in the clean room, for example, in the lab. Not from your skin, obviously, inanimate surface. So it removes it completely or reduces the number of microorganisms to a level compliant with regulations, because not harmful to health or just compliant with specific regulations. If you're using disinfectant in a teaching lab versus using the disinfectant in a clean room in a pharmaceutical facility. The regulations, the expected level of cleanliness, so to say, right, of the surface, those regulations, those expectations will be different. So disinfectant doesn't necessarily always remove all of them completely, right? Sometimes it's just reducing it below a certain value, beyond a certain concentration. But again, the key word is inanimate surface, and it should be pretty self-explanatory, right? I mean, you're all grown-ups. I mean, you've used disinfectants before. Disinfectants are pretty harsh chemicals, as we will look in a second. So applying those to your hands or to any other epithelial tissue in general is obviously a bad idea. It's dangerous and so on. So disinfectant is a harsh chemical. It's the most harsh chemical of these four classes that we will look at. Disinfectants are more harsh chemicals than antiseptics. They are more harsh chemicals than preservatives, obviously, and obviously even more harsh chemicals than antibiotics. Which again should be very logical. Antibiotics are the chemicals you take in, right? You swallow them. So obviously, there's got to be something. There's got to be a big difference between disinfectant and antibiotic with that respect. Um, like I said, we will look at the specific classes later next week. Well, next week, I guess, after the midterm. But in general, all disinfectants and their application can be subdivided into three levels of disinfection. Um, Depending on the application, depending on the needs, depending on the regulation specific to a facility or specific to a particular room or whatever facility you're being in, they distinguish three different levels of disinfection: low, intermediate, and high. And this table shows you what is expected to happen uh, when you apply disinfectant for each disinfection level. So a very logical, get the logic from the table. It's not that complicated at all. The low level of disinfection versus high level of disinfection obviously refers to how many different species of bacteria, viruses, or fungi are expected to be removed. Obviously, low level of disinfection removes less than a high level of disinfection. And these are just the specifics. Know the specifics though. I will go over the table right now. I'll tell you exactly what to remember. So the low level of disinfection expects you to remove and kill most bacteria, right? The key word is most, so not all of them, right? Some may survive, some viruses, some fungi, right? So pretty lenient regulations. Again, sometimes students think, well, every time we use disinfectant, we clean the surface completely. We remove all the microorganisms. Not necessarily. Depends on the level of disinfection that is being applied. The level of disinfection, we'll talk about it later, but different levels of disinfection differ in terms of concentration of disinfectants, as you can imagine, right? The higher the concentration, the higher will be the disinfection level for obvious reasons. And some other factors. We'll talk about that. So with low level of disinfection, you're removing most bacteria, not all of them, some viruses and some fungi. Okay. The key point here is not everything is being killed at low level of disinfection. So what is supposed to survive, or not supposed to survive, I guess the right word, but what can survive, and that would be still acceptable. Um, Bacterial spores can survive. Why is that the case? How do you memorize that for the test? Because bacterial spores are very tough entities, right? Biological entities to deal with. A lot of them, maybe not all of them, but a lot of them. So it is expected that with low level of disinfection, some of those bacterial spores, maybe even most of them, will be able to survive. Some viruses and prions. Viruses can differ significantly in terms of their levels of resistance. Some viruses are easily removed. Some viruses are not so easily removed. So some viruses may survive, and that is acceptable. Like I said, it is understood that at low level of disinfection, some viruses will survive. Prions, as you know, those are misfolded proteins. They're not even considered to be living creatures. Similar You know how with viruses they say that they're not really living because when they're in the host, they show the properties of the living organism, exhibit those properties. When they're outside of living organisms, they're like inorganic forms of life. So with prions, it's even worse, so to say. Prions are just misfolded proteins that cause other proteins that come in contact with them to get misfolded as well. And those misfolded proteins cause all sorts of nasty consequences for the host because, as you know, misfolded protein may lose the functionality. And that loss of functionality because of loss of the shape and therefore loss of the structure of the function rather obviously can cause severe consequences. The mad cow's disease that everybody probably heard by now is being caused by prions. So the prions are just proteins, and they are extremely can be extremely tough to get rid of. There's very few labs that are selected few labs that are even allowed to work with prions, like on a national scale. I'm talking about like in Canada. It's not like any lab can have prions working can be working with prions. It's extremely stringent regulations regarding these. They are not easily removed. This guy here that I left out, Mycobacterium tuberculosis. Mycobacterium tuberculosis. So this is a specific species, obviously, that causes tuberculosis. The bacteria cause tuberculosis, but what you need to also understand, this is kind of like placeholder. It's so to say, it is a symbol, not a symbol, but yeah, I guess a symbol for um, more resistant bacteria, or generally bacteria with higher levels of resistance than normal. There's within bacterial world, there are bacteria with normal levels of resistance and the ones that have elevated levels of resistance, higher than average. So Mycobacterium tuberculosis is being representative of the species, bacterial species that have higher than normal on average level of resistance. So this guy here, this representative here, refers to not just Mycobacterium tuberculosis, but all bacteria that may have higher than normal levels of resistance. Okay, it's just we cannot put obviously every single representative there, but usually species with higher than normal levels of resistance, elevated levels of resistance. So those microorganisms, that bacteria is also expected to survive the low disinfection low. Some may get killed, but some may survive, and it is acceptable. Pseudomonas, for example, species can have elevated resistance. Acinetobacter baumannii can have elevated levels of resistance. E. coli, so for these species, one thing I guess you got to understand, for example, take E. coli. Um, there are certain E. coli species that are very easily removed by disinfectants. They just pose no problem. But within the same species, E. coli, there could be representatives that have elevated levels of resistance. They're still E. coli, but they have elevated levels of resistance. So those species will also, that kind of E. coli will also be down there. Okay, so it's not like all E. coli have the same level of resistance and they go either up here or down there. No, there could be some E. coli with low levels of resistance or normal levels of resistance, and certain E. coli species that, for example, mutated or uptake in the plasmid with antibiotic resistance gene or disinfectant resistance gene that will have higher levels of resistance. So Mycobacterium tuberculosis is just an example of species that have elevated levels of resistance for different reasons. Okay, intermediate levels of resistance. So here, what you're removing is what you're expected to remove. Most bacteria, including these, not just Mycobacterium tuberculosis, but any bacterial species with higher levels of resistance. So intermediate already implies that most bacteria should be removed. Okay, except for why most. So what are the exceptions? Some may ask. Exceptions will always be there, there always could be some species with exceptionally high levels of resistance, and it still applies even for the high level of resistance. You're expected to remove all microorganisms unless you have some extremely resistant bacteria. That can always happen. Some absolutely unexpected species that have extreme levels of resistance. So there will always be these exceptions, or might be these exceptions, but technically most bacteria should be removed. In certain cases, you cannot account for every single scenario. So there might be some exceptional species out there that will still be left untouched by the disinfectant. Most viruses, including some of the more resistant viruses like hepatitis B, so same deal. Most viruses, except for some, uh, again, rare exceptions. Most fungi, right? Bacterial spores still expected to survive. Again, maybe not all the spores because obviously tons of spores at different levels of resistance, but some spores may get removed, but it is okay. It is expected that bacterial spores will be surviving intermediate level of disinfection. Prions, like I said, some of the most resistant species, biological entities we have. Um, they will survive even high level of disinfection, as you can see here in the other part of the table. So the high level disinfection technically supposed to remove everything, right? Except for those, like I said, um, unique extreme examples, just outliers, right? Some extreme challenge, uh, certain resistant bacteria can pose extreme challenge. Usually that kind of bacterial species are making it into the news. Doesn't happen often, but uh, can happen. The spores, same deal, extreme challenge of resistant bacterial spores, like super resistant bacterial spores. But technically, everything has to be removed. Um, with some, in microbiology, there's always going to be exceptions. If you're lucky, that in that particular case, no exceptions will be. You will not face any exceptions, but there's always a chance that you will come across something, right? Come across something that may fall out of this table and be resistant. Prions still expected to survive certain levels of disinfection because prions are just extremely tough structures to get rid of. What I guess so pretty self-explanatory table. Um, what constitutes, I guess, what governs the level of disinfection? So how do we know if the level is low, intermediate, or high? So first of all, what kind of factors impact the level of disinfection? The level of disinfection. Obviously, the identity of um, disinfectant, right? Um, certain chemicals are more harsh, certain chemicals are less harsh. We all know that, I don't know, rubbing alcohol does have disinfectant properties, but bleach, household bleach, obviously is a much stronger disinfectant, right? To give you an example. So identity, chemical nature of the agent, concentration between different agents or even within the same agent. Super low concentration of bleach has probably, well, not probably, but definitely has less of an impact than a high concentration of bleach. And same applies to any chemical. Make sure you understand, again, may sound basic, may sound obvious, but disinfection is a chemical reaction. Um, you know, it doesn't happen the way they show you in commercials. I hope everybody understand that. Where you pour disinfectant and poor bacteria just start exploding or something like that into nothingness, into the void or something like that. It's a chemical reaction. You pour, you expose the bacteria to the disinfectant, which is usually an oxidative agent. So as we will discuss later in details next week, uh, most disinfectants act on bacteria by oxidizing their cell wall and the cell membrane. So they're oxidative agents that can, as you know, should know from chemistry, they are very hungry for the electrons. That's what the oxidative agents are, right? And they oxidize cell membrane of the bacterial cell. As they oxidize the cell membrane loses the integrity, and then obviously the cell loses the integrity. Everything is spilled into the environment. You kill the bacteria, you lyse the cell. So if it's a chemical reaction, that chemical reaction is subject to all those parameters that any chemical reaction, is subject to, right? Concentration, that is, exposure time. So the identity of the chemical you're dealing with, because different chemicals have different mode of action depending on their and different efficiency depending on their chemical structure, right? So keep that in mind. So concentration, obviously, and then I already mentioned that exposure time, a factor very commonly um, overlooked, I guess, by students as well, which is not that big of a deal, I guess, in teaching labs. But when you pour the disinfectant on the surface of the bench in the lab, a lot of people just wipe the bench down immediately. You got to understand that in order for the chemical reaction to happen, in order for the disinfectant to kill bacteria, it needs to be exposed to that bacteria for some time, because it's, like I said, it's a chemical reaction. It has a certain rate. It has a certain, uh, I guess, efficiency. So when you pour the disinfectant, you have to let the disinfectant uh, get exposed to bacterial cells for some time. So give it some time, 30 seconds, a minute or so, and only then wipe the bench down dry. Don't do it right away. So, and if you might remember from what the lab instructors told you at the start of the course, when you spill the culture on the floor in the lab, you're supposed to pour the disinfectant into that spill and let it sit for, like, I think the regulation is like half an hour or something along those lines, 15 minutes or half an hour, right? So why are we exposing it for so long? To allow the disinfectant to completely kill bacteria, right? To allow for that chemical reaction to go through in its completeness. So exposure time is huge. Um, there are more efficient disinfectants, like very efficient very harsh disinfectants that act very quickly. But there are also bacteria that are very resistant, can be very resistant. So you never know what kind of bacteria you're dealing with. That's why there's always, by default, you should be sticking to the regulations and exposing your disinfectant to the bacteria for an allocated time, whatever it is. Yeah. Assuming when you expose your bacteria to the to disinfectant, you kind of have to assume that you're dealing with resistant species, even though you may not be, but you have to assume that because you never know. So exposure time. I guess these are the major ones. The fourth, I guess, major one would be the temperature. We'll look at it in a second as one of the factors, the temperature of disinfectants. For obvious reasons. Again, if it's a chemical reaction, the temperature of the chemical reaction, as you should know, does impact the rate of the chemical reaction. And even if you think about it, like, kind of layman terms, um, boiling something in disinfectant is probably going to be more efficient than just exposing it to a cold disinfectant. And it's generally the case, unless obviously the disinfectant gets chemically gets destroyed, right? It gets decomposed at high temperature. So obviously, if that's the case, then you should be using it cold. But if it is stable at a certain temperature, the higher the temperature, the more efficient the reaction is going to be. So temperature would be the big one as well. So that's that. And as we'll also look at one more, I guess, the pH. pH is also like often overlooked. People, even in the lab, sometimes people, when you guys do the project, I remember from the past when I had a few project sections a long time ago, people kind of overlook the pH factor in general, not just for disinfection. You're not really dealing with pH during disinfection, but when you guys set up all sorts of different reactions, people forget that pH may have a drastic impact on their reactions. A very common mistake I remember was where people expect a certain color to develop in their reaction, and they don't adjust the pH properly. Color never develops because pH has been never adjusted. So keep that in mind. pH is just as much of a factor as anything else in color development, or in our case, in disinfection. pH is a factor, right, that affects the charge, the level of protonation of chemicals. So obviously, if it affects something, it may affect the outcome. So pH is important, and I will show you how it is important today, and we'll talk about those. So those are the factors. We will look at all of them, I think, or most of them. Uh, with all of them, I guess, within the scope of the whole lecture, but some of them only today, in more details or less details, depending on the factor. Anyway, antiseptics, the next chemical class of chemicals. Very briefly, we're not going to spend much time, any time really, about on antiseptics. We will kind of talk about them when we talk about specific disinfectants, because a lot of chemicals can be used as both disinfectants and antiseptics. It's just a question of concentration quite often. So antiseptic, make sure you understand what it is. It's an agent. Literally, that translates something against infection. Antiseptic. That will kill or inhibit organisms on a living tissue. The key word here is a living tissue. So that's something you apply to your hands, right? The first thing that should come to your mind. You should not apply disinfectant to your hands for obvious reasons. It's a harsh chemical. Antiseptic is specifically being used, formulated, and used for living tissue. Surgeons use antiseptics when they disinfect. They use for, I guess, living tissue, they use antiseptics. For instruments, they use disinfectants. So it's not a synonym to disinfection. For disinfection, it is a different type of agent, different application, I guess, of an agent. It could be the same agent, but for example, alcohol, like ethanol, can be used as a disinfectant as well as antiseptic, the same chemical. The question is, what is the concentration? What is the preparation of that chemical? So, but key point is it should be safe to apply to skin or mucous membranes in general, right? So that's what surgeons do. Applied before surgery. And like I said earlier, the same agent can be used as disinfectant and antiseptic. Not all of them. There are certain chemicals, for example, like peroxides. Well, peroxide actually can be used as antiseptic. Certain types of peroxides, more not necessarily hydrogen peroxide, but more active peroxides cannot be used as antiseptics, but quite a few chemicals can be used. Like bleach, obviously, should not be used as an antiseptic. But certain ones like rubbing alcohol can be used both. Ethanol can be used as both, and whenever the chemical is being used as both disinfectant and antiseptic, the difference would be in concentration of the agent for obvious reasons. The higher the concentration, generally, the more effective is the chemical. So if you were to go about the alcohol, the higher the concentration, the more likely it will be it will act as a disinfectant. The lower the concentration of alcohol, the likely it will be used as an antiseptic. Even if you lower the alcohol concentration even more, it can be also used as a preservative, as you know, especially in certain type of products. I think in certain cosmetic products, I believe they use alcohols as preservatives, which is the next class we will look at. So the concentration, the concentration of the chemical is lower for the antiseptics. It's higher for disinfectants, lower for antiseptics. Preservatives, the next class, we will briefly touch upon that. Something you did in the lab 2 weeks ago, I guess, before the break, where you were testing the effect of the preservatives. It's the chemical specifically added to the products to extend the shelf life and prevent the growth of microorganisms. Technically, all preservatives should be preventing the growth. I want to say this should be a key word here, but nowadays preservatives can be so efficient that they don't just prevent the growth, but they also kill bacteria. So they have both bacteriostatic as well as bactericidal effect. Those of you who don't know, I think I actually have this. Yeah, I have it on the next slide. Bactericidal is the chemical that kills bacteria. Cidal, right, killing. Bacteriostatic means it is just prevents the reproduction of bacteria, so it keeps the concentration unchanged. Okay, static. That hence what static is for here. So technically, preservatives should be all static. That's how they were initially, but nowadays preservatives and the chemicals that they use for the preservatives, they can actually kill bacteria as well. So they can reduce the concentration of bacteria, not just keep it in check, but also reduce it by killing it. And it's obviously, since it's added to the products, the preservative should be safe for intended consumption of the product, right? If the preservative is being added to a food product, the preservative should be safe for ingestion, right? For when people eat the product. If it's a cosmetic product, it should be safe for the skin and whatnot. So that's consumer use here is key. So it's non-toxic for intended route of administration, obviously. Technically just inhibition, so of microbial proliferation, but quite often, like I said, they are cidal as well. Out of these three classes that we looked at, disinfectants, antiseptics, and preservatives have the lowest concentration of the agent out of the three, the lowest. So in my example that I gave you earlier, alcohol, quite often different alcohols are being used as can be used as three different applications as disinfectants, antiseptics, preservatives. Preservatives obviously will have alcohol used as a preservative will have the lowest concentration. It's not even, it's just a kind of should be pretty obvious just by looking at the application, judging by the application of the chemical. Obviously, a lot of preservatives are in the food nowadays, especially they add it to all sorts of. It's hard to imagine a product without any preservatives added, right? So obviously, it has to be safe. Some of these terms, this is just the basic definitions. I mean, in no particular order, just make sure you understand what these mean. Most people are familiar with these, but not always. Bactericidal means the agent that kills bacteria. Doesn't matter what kind of agent, as long as it kills. Or could be a bactericidal effect, for example, right? So the chemical shows a bactericidal effect, meaning that the chemical kills bacteria. Bacteriostatic means it just prevents the replication of bacteria, so keeps the numbers in check, but doesn't kill. It doesn't reduce the concentration of bacteria. Okay. For example, a lot of antibiotics, the class of chemicals we will look separately in one of the last lectures of the course. A lot of antibiotics, the ones that you are getting prescribed when you get an infection, a lot of people think that they actually kill bacteria. Most antibiotics actually don't. Most antibiotics have bacteriostatic effect on bacteria. They just stop the proliferation, and then your immune system takes care of whatever existent infection you have. There are also bactericidal antibiotics, don't get me wrong, but most that you're using, unless you're facing some severe condition, most antibiotics are bacteriostatic. Okay, keep that in mind. Detergent, very common actually misconception, or used to be, hopefully not the case anymore. I don't know, hard for me to say. A lot of people think detergent kills bacteria. It doesn't. It just reduces the surface tension and doesn't really kill anything. Classic example is soap. Even though this example requires some clarification. Soap, just the regular soap, doesn't kill bacteria, it just washes it off your hands. But nowadays they sell so-called bactericidal soap, and that's what people like to purchase often. They sell them everywhere, right? Bactericidal soap. Well, bactericidal soap does kill bacteria, but it's not the soap that kills it. It's the chemical, specific chemical called triclosan, usually, that kills it, that is being artificially, so to say, added to the soap. The soap itself doesn't. So that's one thing. The second thing about that soap, since we will talk about this more later in the course, but um, I'll explain in more details. You should not be, if you ask me, and anybody who understands a little bit behind the science behind these things, you should not be using bactericidal soap. It's not a very smart strategy. It's not it's not super dangerous per se. Like don't panic. But um, whenever you use bactericidal soap, you kill bacteria or some of it. What happens when you kill bacteria? You promote the evolution of bacteria, and you promote, therefore, their development of resistance. Um, when this kind of soap is being used en masse by millions of people, you imagine what happens to bacterial communities that are getting exposed to this soap. What happens to bacteria in soil that gets exposed to this runoff, to these runoffs, the water that comes from the households where people are using the bactericidal soap. You're promoting resistance. We as a society, by using that soap, are promoting resistance. And if you think there's some sort of conspiracy or something like that, it's not. If you talk to actual scientists uh, who are in the know, I guess, because sometimes people work in other fields, they have no idea about, for example, antibiotics. But people dealing with antibiotics and bacteria, any smart people, I guess, will tell you that it's a horrible idea. And um, there were some time ago, I'm not sure where they are standing with this at this point, but some time ago, there was even I knew people, the scientists in Canada, who were actually in talks with government trying to ban this soap. There's overwhelming evidence that it's not a very smart um, idea to to sell that kind of a soap to general public because of the resistance, because we're promoting resistance. And then you hear about all these nasty species in the hospitals that kill people because they're so resistant. So by using antimicrobial soap, we actually promote that resistance. It's not a smart choice. So keep that in mind. Why do they sell it? Some may ask. Well, the answer is pretty straightforward. It sells. Most people obviously are pretty far, like far away from science, so to say. They don't know much about these things. They just why wouldn't I use the soap that kills bacteria? It's so good, right? So people buy it, and they don't really understand all the reasons, pros and cons, so to say. But it sells, and obviously brings profits and whatnot. But it's not a very smart choice, especially when it's being used like by millions of people, and we expose bacterial communities to additional uh, stress factor that causes them to mutate, that makes them mutate. So in states, I know, I'm not sure about Canada, maybe it's also the case in Canada, but in states, they even sell sportswear soaked in triclosan, like socks, like gym gloves, like those kind of things soaked in triclosan. And they market it as, it's just, to protect your workout so that you feel better, you feel safer. So, and people buy it because they think, well, yeah, I want to protect myself, right? So, but it's not a it's not a very smart choice at all. Not to mention that you're exposing your skin to a chemical for no real reason. No real reason. After all, your immune system, unless you have a certain condition, there's also obviously immunocompromised people. It's a different story. But the general public, healthy individuals, after all, you want to train your immune system. You don't want to rely on the chemicals constantly taking care of bacteria, fungi, viruses, and everything else, right? So. But anyway, we'll talk about this much more when we talk about the antibiotics in the future. But fungicides, the chemicals, the agents that kill fungi, Obviously, as the name suggests, germicide is more of I don't want to say non-scientific, but it's more like a term from newspapers, more so than scientific term. Basically, the agent that kills pathogenic microorganisms, chemical that kills the germs, right? The germs are not necessarily a very scientific term. I don't know. Scientific literature usually uh, tries not to use this kind of term. Bactericidal, bacteriostatic, yes. Germicide, I guess not so much. Sanitizer, the chemical that reduces the agent that reduces the level of microorganism to a level that is considered safe, right, by public health, for public health. So that depends on the application. So different requirements are different for different settings. One thing you're dealing with food industry, the other thing you're dealing somewhere. I don't know, uh, some sort of a other facility. So sanitizer just reduces the level of microorganism to a level considered to be safe for public health. Um, Sterilant, specifically the chemicals that removes, kills all forms of microorganisms, including spores. Again, ideally it does so. Keep in mind, nothing is ever black and white. It's not like there's a chemical that kills everything 100%. There's no such thing. Sometimes it will kill everything. The other times it won't. Um, so sterilant, as the name suggests, the easy way to memorize this, uh, the agent that sterilizes, right? Sterilant, something that sterilizes. So what is sterilization? Complete removal of everything, all the microorganisms, making your surface or whatever have you sterile, right? So that's what sterilant is. So those are some basic definitions you have to keep in mind. So as far as the choice of antimicrobial and all these factors that govern as I was showing you here, the level of disinfection. So let's talk about all of these factors. Um, what this actually, yeah, let's actually cover this as well, and then we'll talk about different factors that impact the activity of disinfectants and therefore your choice of those disinfectants. So some more definitions or some more terms. Again, probably familiar to you. You must have performed the MICs and MBCs in your project courses. Project One, I remember back in the day, um, we had a lab where people were setting up MICs. So MIC and MBC, what does it stand for? This stands for minimal inhibitory concentration, minimal bactericidal concentration. This refers to concentrations, as the title suggests, the minimal concentrations of an agent that inhibits the proliferation of bacteria. Okay, minimal, uh, because if you crank up the concentration, you will probably keep inhibiting bacteria. So MIC specifically talks about the minimal, the first value, so to say, the minimal value that does so. That is your MIC. MBC would be the minimal value that kills bacteria. So these parameters are being used quite often in the lab when people estimate the efficiency of an agent, this or that agent. They compare the MIC for alcohol, for example, and the MIC for aldehyde. And then they make a decision which chemical is more active. Obviously, the lower the value, the more active is the compound. If it takes you, I don't know, 1 milligram per liter versus 10 milligrams per liter to inhibit bacteria, which one is more effective? The one that takes less. That one is more effective. So know what they stand for. MFC stands for minimal fungicidal concentration. So that's about fungi, but the same concept. Also, make sure you understand that MBC is usually higher, well, than MIC because concentration to kill will be higher than concentration to inhibit because it's a more, I guess, I don't know, more harsh, I guess, application for the chemical to kill a bacteria. So MIC is lower, MBC is higher. You first inhibit bacteria, and as you keep cranking up the concentration of that chemical, you eventually kill bacteria, right? So MBC will always be higher. But again, it's minimal bactericidal concentration. So the what's the minimal concentration of the chemical of this chemical that I need to kill bacteria? 10 milligrams, for example, per liter may kill, but 20 will also kill, 50 will also kill. You're looking for the minimal. We will talk about that in a second. Why are we looking for a minimal concentration? Because we don't want to overexpose our bacteria, our microorganism to the chemical. Why we don't want to overexpose? Because we promote resistance. We want to use the minimal, most effective concentration at all times. And if bacteria becomes resistant to it, then you can elevate the concentration and say, okay, can you handle this now? Right? So you always start with the minimal concentration. It's like, I don't know, maybe not a very uh, proper analogy here. The first one pop in my head. It's like betting, right? I don't know, somewhere in casino or something. You don't bet maximum right away, right? You bet a little bit, like minimal value you possibly can, and then see how everybody else does. So the same kind of deal here. You don't want to be exposing your bacteria right away to the highest concentration. Um, Tolerance is the ability of the microorganism to survive uh, under normally what is supposed to be cidal concentration. Uh, Bacteria may survive that cidal concentration, but will not replicate. And if bacteria shows that behavior, they say that the bacteria shows tolerance. So what does that mean? For example, that 10 milligrams per liter of a certain agent should kill your E. coli. You apply 10 milligrams per liter and you realize bacteria actually doesn't get killed. It gets inhibited. So what does that imply? That implies that this bacteria shows tolerance. In other words, it's just another way to indicate that bacteria starts showing elevated levels of resistance to that chemical. Something that is supposed to kill it all of a sudden doesn't kill it anymore. Inhibits it, but doesn't kill it. So tolerance is like another word for low levels of resistance, right? Now on to the factors, actual factors, like I said, the ones that govern the choice of antimicrobial and the ones that impact the efficiency of disinfectants, as well as antimicrobials in general, most of them. So when I was giving you this list a few slides ago, I started with nature of the chemical, right? The identity of the chemical. So that one we will cover at the end of the lecture. We will go over a bunch of classes of chemicals like alcohols, peroxides, aldehydes, and so on and so forth. So that is going to be at the end because it's not a part of the second midterm. So we will talk about that later. Concentration. As far as the concentration, I don't think I actually have a separate slide here. Concentration is an obvious one. Like I said, the higher the concentration, the more efficient is the agent. Okay? Maybe make a note on the side. There are some, I don't want to say exceptions, but there are some, um, I guess, interesting cases here that might happen. Sometimes, um, as you elevate the concentration, the dependence is not that linear, let's put it this way. So sometimes when you elevate the concentration to a certain extent, the efficiency of disinfectant increases. And then at some point, it actually starts dropping or remains flat and then drops. So we will look at those examples next week, not this week. But just those are kind of like exceptions to the rule. Let's put it that way. Generally, the higher the concentration, the more efficient is the agent, obviously, because it's a chemical reaction. The higher the concentration, the higher is the rate of the reaction. Temperature. So another one that I gave you out of the four or whatever it was, temperature. Temperature, again, the higher the concentration, the higher the temperature, the higher will be the rate of the chemical reaction. Unless obviously you heat up the disinfectant beyond its stability point. So this is just something to keep in mind. Different disinfectants have different stability. So obviously, if a disinfectant gets decomposed at boiling temperature, you should not be exposing your surface to that temperature. So just a technical, you got to know those technical details about the disinfectant when you apply it. But generally, with elevation of temperature within those safe limits, Elevation of temperature leads to increase in efficiency of disinfectant. As it says here, the cytal activity rises as temperature increases. How do people quantify it? There's two coefficients you need to know. The one is called temperature coefficient, designated by the Greek letter theta. This guy here, theta, which shows you the change in efficiency of disinfectant per every 1 degree Celsius, per 1 degree rise of temperature. This coefficient is not very useful because 1 degree Celsius is not necessarily a very commonly, like people increase the temperature usually by more than 1 degree Celsius. So instead of theta, people quite often use the so-called Q10. They don't contradict each other. This one, in no way. It's just the Q10 is a bit more convenient to use for application anyway. For research purposes, you might want to, stick with theta and track the change in efficiency per every 1 degree. But for the application, people quite often stick to Q10 coefficient. And this one, very similar. I mean, it's an identical concept to the theta, except for this one shows the change in efficiency of disinfectant per every 10 degrees Celsius rise of temperature. Okay, so just 10 degrees. How it is calculated, you must know this formula for the test. It is a simple formula. You take time to kill at a certain temperature, which is your baseline, let's say 20 degrees Celsius, and you divide it by the time it takes the same disinfectant to kill at T plus 10, which is the temperature that is 10 degrees higher. That's it in Celsius. So time to kill at T, say 20, you're comparing the efficiency of disinfectant at 20 versus 30 degrees Celsius. So you expose your bacteria to this disinfectant at 20 degrees, and you measure the time it takes to kill all the bacteria. And then you do the same thing at 30 degrees Celsius, and you measure the time it takes to kill. And then you just divide the first one by the second one. So if it took you 10 seconds to kill all the bacteria at 20, and it took you 2 seconds to kill at 30, 10 divided by 2 gives you 5. So the Q10 is 5, meaning that your disinfectant becomes 5 times more efficient for every 10 degrees Celsius elevation of temperature. Pretty straightforward. Just one number divided by the other. Okay. What you will need to know for the test also, being able to compare, super straightforward, the Q10 coefficients for different chemicals. So here's an example. Phenol has a Q10 of 4. So first of all, make sure you understand what it means. It means that phenol becomes 4 times more efficient for every 10 degrees Celsius temperature elevation. What does it mean 4 times more efficient? You refer to the formula. It kills 4 times faster for every 10 degrees Celsius of temperature elevation. Okay. Another chemical, let's say ethanol, has a Q10 of 45, so it kills 45 times faster, right? Significantly faster for every 10 degrees. Ethylene glycol ether has a Q10 of 300. It kills 300 times faster for every 10 degrees Celsius. So if I gave you these values or some other values and ask you which one is scales more efficiently with temperature, obviously it's the last one, the ether, right? Because its efficiency jumps faster, 300-fold every 10 degrees Celsius. Which one is the least efficient with respect to the temperature elevation? Phenol in this example we're looking at. It's pretty straightforward. Usually the efficiency is standardized at standard condition, which is 20 degrees Celsius. And then you elevate the temperature from 20 to 30 to 40 to 50 to 60 until as long as the disinfectant is stable. Don't forget, some disinfectants, considering that they are such reactive species, chemical species, those kind of species are usually not so stable with temperature. They just start losing their structure. So you cannot just assume that your disinfectant will be always stable. Even at like 50, 60 degrees Celsius, they might start decomposing. So when you work with disinfectants, refer to the MSDS sheets, refer to the safety data sheets, that is, refer to technical characteristics of the chemical, know its stability. So that's pretty much I guess the logic behind the temperature. There's nothing complicated about that. pH. As far as pH is concerned, let me stop sharing the screen for a second. Let me open the Microsoft Word, and I'll just I have a file from before. I have them saved by now, but it's a minimal amount of text, so it's not a pH requires a little bit of explanation. So I'll show it to you. As long as you understand basic chemistry, really basic chemistry, you should be fine. I'll explain it anyway. I'm sharing the screen now. So how does pH impact the efficiency of disinfectants? First of all, in order to understand this concept, you need to remember what pH is. pH, as it shows here, let me zoom in a little bit more, is the negative logarithm of concentration of H plus ions in your solution. H plus ions, the measure of acidity. As you know, negative logarithm. So you take the logarithm of log 10 of the H plus concentration and take it as a negative value. That is your pH. You should remember these ranges, which environment is considered to be acidic, which one is considered to be basic, which one is considered to be neutral. So 7 is neutral, less than 7 is acidic, more than 7 is basic. Why is it such, if you noticed a little bit counterintuitive pattern, I guess, why is it acidic if pH is less than 7? Because you take negative logarithm. So the higher the concentration, the lower is going to be the pH. So that is a little bit counterintuitive. So pH of 1 is a very acidic environment. pH of 10 is very non-acidic environment. It's a basic environment. It's counterintuitive because of the negative sign here. It's the opposite. High concentration of H plus will give you a low pH value. So again, that's chemistry. As far as the impact of pH on activity of disinfectants. So most disinfectants, not all of them, but a lot of them, are so-called weak acids. That's their chemical characteristic. You should have studied this at some point, what the weak acids are, but I will revise it. So what is a weak acid? So first of all, acid is the chemical, right, that is capable of donating protons in the solution, right? So generally, acids in chemistry are like a general notation for the acids is HA, like this. It's a general notation. It doesn't refer to any specific acid, just acid as in general. So H stands for obviously the proton. A is your anion. So for example, HCl, H is H, anion is the chloride ion in your HCl. So HA is a general form, general notation for any acid. So sulfuric acid, you have H2SO4, right? So SO4 2 minus is your anion. So weak acid, what is a weak acid? In order to understand the impact of the pH on the activity of disinfectants, you need to remember what a weak acid is. Any acid in the solution, but because of its nature, what it does, like I said earlier, it donates the protons. So it can dissociate, right? Dissociate from its undissociated form into H plus ion and anion, whatever that anion is, chloride or chloride or whatever, NO3 or SO4, any of those. OCl for the bleach, HOCl is the acid, OCl is the anion. So it can dissociate from its undissociated form into proton, right, and the anion. Um, Forget about the OH on the right here for now. So a strong acid would dissociate fully, right? This process will go from left to right fully. Weak acid, the ones that we're looking at in the context of disinfectants, this dissociation process can go both ways. So the acid dissociates into ions, but at the same time, in the solution, it's a dynamic solution, dynamic balance. The ions that are generated, these two ions that are generated by the dissociation, can also come together and form undissociated molecule HA again. Okay? So any weak acid is in this kind of a balance in the solution. It exists in both of these forms, and there's a certain ratio between these forms. There's a certain balance between these two forms. And that's why when they write it down in chemistry, they put these arrows back and forth, right? You know what I'm talking about. Left, the arrow going from left to right, and then the other arrow underneath going from right to left, implying that the process can go both ways. So, in order to remember, uh, in order to remember like how does pH affect those disinfectants, like weak acid disinfectants, you need to remember one thing, that disinfectants that are weak acids, as it says here, are most active towards bacteria, that is, when they are in their undissociated form. Undissociated form. So in this form here, that's where their most bactericidal activity is. So uh, our question gets to the point where you have to ask yourself, so if this equation is in balance like this in the solution, how do we ensure that um, um, our weak acid is um, present in the solution mostly in undissociated form? How can we alter the solution in such a way that that our acid is present in H plus undissociated form? So in other words, as they would say in chemistry, how do you shift, right, the balance of this equation to the left, right? So in order to remember that, you just have to remember the basic chemistry. Um, if you were to add to your solution of your disinfectant some more H plus ions, right? What's going to happen? If you add H plus ions, so if you place your disinfectant at low pH, right? If you lower the pH of the solution of your disinfectant, you will have an abundance of H plus ions on the right-hand side of this equation. And because of this abundance, you will have the reverse reaction going more often than the forward reaction. In other words, your weak acid disinfectant will be dissociating less than it will be, you know, associating, these ions will be associating back together to form H plus. So by supplying extra H plus to the solution of your weak acid disinfectant, you are shifting the balance of this reaction from right to left. And that means you are promoting, I guess, higher concentration of an undissociated form of the disinfectant in the solution. Okay? Not all of the weak acid will remain in undissociated form, but most of it will. And that means in order to enhance um, the efficiency, antimicrobial efficiency of disinfectant like this, you have to use this disinfectant at low pH in acidic environment, as it says here, because you are shifting the balance to the left this way, and this is the more active form of disinfectant. The more active of these two. Undissociated is more active than dissociated. That's why you want to shift it to the left. Okay. If you were to do the opposite, that's what I have the hydroxyl ion here on the right. If you were to do the opposite, if you were to supply your disinfectant in a basic environment by supplying additional OH minus ions, right? Hydroxyl ions, what's going to happen? OH minus will react with H plus, as you should know from chemistry, and form very thermodynamically stable molecule that is water, H2O. And as you do that, you remove H plus from the solution. It pairs up with OH and forms water. As you remove H plus from the solution, you promote further dissociation of HA, of your molecular form, I guess, of disinfectant. And as you keep promoting that dissociation, you have progressively fewer and fewer acid present in undissociated form. And that's the best form. You don't want this to happen. So that kind of confirms what we just talked. So if you were to place the disinfectant into basic environment, you would lose your undissociated form, and therefore you would lose the activity of antimicrobial activity of disinfectant. So for weak acids, it's H plus. You want the low pH solution for that disinfectant. There are also disinfectants that are weak bases. They're less numerous, but they exist. So for the weak bases, and you can do it on your own, apply the same logic, like literally the same logic, except for the base will dissociate into cation and OH minus. So for the weak bases, you actually want to use them in basic environment for the same reason, because undissociated form of the weak base is the most active form of disinfectant. So for weak acids, it's low pH. For weak bases, it's high pH. Okay, so that's pretty much the logic behind all of this. Let's go back to the lecture slides. Okay. So yeah, the pH, I guess some general considerations, what pH may affect, what we just looked at. You can put this first, doesn't matter. It may affect the degree of ionization of the disinfectant. What we just looked at was the degree of ionization. HA is non-ionized form of disinfectant. H plus plus A minus is your ionized form. So pH affects that level of ionization. At low pH, you have it more in deionized form. At high pH, you will have your weak acid at ionized form. So as we just discussed, you want it to be in ionized. So strong acids can never act as disinfectants. No, they can act as disinfectants. It's just strong acids are very harsh chemicals. Very harsh chemicals. They react with surfaces and whatnot, so they're not necessarily the best choice. And when you, that was a question. Sorry, not everybody probably sees that. Basima asked the question. Another thing is. The acid when you use a chemical as a disinfectant, its strength as an acid or the base is not necessarily what you're looking for your application. You are looking for antimicrobial activity. And antimicrobial activity and the strength of the acid or the base are not the same thing. Don't confuse these two. Okay? So the chemical should be able to oxidize. It should be a strong oxidizing agent. That's what the disinfectant is. Not the strong acids quite often are not actually strong oxidizers. Not always they are. So for that reason, there's no correlation here. Okay. You want the ability to oxidize as opposed to ability to be a strong acid or a strong base. Okay. So usually weak acids are stronger, better disinfectants. If you were to generalize with some exceptions, but weak acids are stronger disinfectants. pH may also affect, so in addition to degree of ionization that we just covered in the doc file, pH may also affect the stability of disinfectant, so you also have to keep that in mind. It may affect the ability of disinfectant to combine with the cell surface sites. So this one requires a little bit of explanation. So like I told you earlier, any disinfection process is a chemical reaction. For this chemical reaction to occur, your disinfectant must come in contact with bacteria and quite often attach to a cell wall of or the cell membrane rather of bacteria. So that attachment is not, so to say, it's not also an automatic process. It depends on charges of the disinfectant as well as the charges on the, like chemical charges, plus minus charges, electrostatic charges, I guess, on the cell membrane. So, and we will look at that in more details next week. So, if that interaction doesn't take place, if your disinfectant is unable to attach to the surface of the cell, that disinfectant may not have any impact on bacterial cell because of that, right? It's like a famous key lock analogy. If you have a key, but if you cannot stick it into your lock, you cannot unlock it. So the same with disinfectant quite often. So in some cases where disinfectants rely on their charge to, in order to attach to the surface of the cell, pH may significantly mediate that attachment because pH may significantly change the charge of the cell surface sites as well as the charge of the disinfectant. So in that case, you have to watch out. You have to just know what we're not going to study that. We're not going to look into any, like, there's nothing really to study there. It's just the same deal. pH, you got to look at how pH changes the charge of the disinfectant and what kind of charge does it require for that disinfectant to be active. We will look at some examples in 2 weeks from now. But the point is that the pH may affect the ability of disinfectant to dock onto the cell surface. And without that docking, without that attachment, disinfectant, regardless of its actual antimicrobial activity, will not be able to oxidize the cell. Another factor that the pH may impact, like a little bit of a, I guess, not a immediate factor, not an immediate, I guess, yeah, factor, but secondary. pH may affect the survivability, the rate of growth, I guess, as well as the resistance of microorganisms. pH may affect the activity of different ion channels in bacterial cell, and that can have a secondary impact on all of these parameters of bacterial cell, right? As you know, there's a lot of channels, ionic channels in the bacterial cell membrane, and those ionic channels play an extremely important role in the survivability, in the activity of different proteins in bacteria. Energy harvesting rate depends heavily on pH. So by changing the pH, you actually may impact these parameters in bacteria. As we will learn later in the course, some bacteria also have these pumps, like literally pumps that in the cell membrane that, just like any pump, can pump out chemicals from the cytoplasm into the environment, and therefore reduce the concentration of those chemicals in the cytoplasm. And as you can guess, some of those chemicals that the bacteria can pump out are disinfectants and antibiotics. So if you change your pH of the solution with this kind of bacteria, that pH, the change in pH, may significantly affect the rate of activity of those pumps. And the rate of activity of those pumps, in its turn, will impact the level of resistance of the bacteria. So those kind of things you also have to keep in mind. We're not going to go into the specifics, but just keep in mind it's kind of indirect impact of pH, right? By just impacting when pH affects some basic parameters of the bacterial cell, like survivability, rate of growth, energy harvesting rate, levels of resistance, and so on and so forth. Levels of communication, biofilm formation can be affected also by pH, like those kind of things. Um, Degree of ionization we discussed. That's what I was explaining to you in the separate, like in the Word file. Survival rate of growth. Okay, we mentioned that. One more factor that pH may also indirectly impact is um, availability of all sorts of other cations and organic material. And cations, anions, all sorts of other charged chemical entities. And some of those chemical entities may have also an indirect impact on the efficiency of disinfectant by affecting these three: survivability, rate of growth, and resistance. To give you an example, you probably studied this in BI 312. You know how magnesium ions, the ones that you use in PCR reaction, are essential for the activity of the PCR, right? For the PCR reaction. Why? Because they impact the activity of the DNA polymerase. So there are also different enzymes that bacterial cell produces that also rely on magnesium ions and calcium ions in its activity. Um, and some of those enzymes are actually responsible for uh, are involved in decomposition of uh, disinfectants. So bacteria secretes those enzymes into the media, and they can significantly reduce the activity of disinfectants or antibiotics. So presence of those ions, like calcium and magnesium, will in the solution will significantly uh, change the activity of those enzymes. So by changing pH, you can reduce availability of those ions, calcium and magnesium. And by doing so, you are impacting the activity of the enzymes that, in its turn, are decomposing disinfectants. And ultimately, obviously, you affect the activity of disinfectants. So certain other ions or certain other molecules can, for example, be used by bacteria to communicate with each other uh, in the form of a biofilm or before the biofilm is formed, as you know. So again, changing pH can actually affect the rate of communication between bacteria, and that can also impact the ability of bacteria to form biofilms. And formation of biofilms will obviously impact the ability of bacteria to survive your disinfectant. So it's a very obviously complex system, especially when you talk, colonies of like communities of bacteria, not just single cells. So pH is a very important factor, often overlooked, like I told you, but it's one of those subtle factors that impacts quite a few things, quite a few things. All right, so keep that in mind. Alrighty, it's been what, over an hour. Let's make a break. Come back at 9 in 7 minutes, as always, and continue the lecture. So 9 p.m. All right, let's get back. Um, so some more factors, environmental factors, I guess, that can affect the activity and the choice of disinfectant. One of them being organic matter contamination of the surface. So in other words, uh, dirt, organic or inorganic, some sort of a dirt contaminating your surface can actually have pretty drastic impacts on the capacity, antimicrobial capacity of the agent for obvious reasons. Dirt can absorb the disinfectant that you apply to the surface. Absorbing disinfectant implies obviously the reduction of concentration. Effective concentration of disinfectant drops. Think of the dirt in this aspect um, as a sponge, right? It just absorbs the chemical and reduces the exposure of the bacteria to that chemical. It can serve as a barrier, as it says, so kind of similar, right? Function. It can just physically protect, the shield off, if you will, bacteria from the chemical that you apply. And it can also block those binding sites that I was talking about before the break, disinfectant requiring certain binding sites on the cell membrane to attach to before it can exert its effect. For example, the citrus disinfectant that you use in the lab, it kind of relies heavily on those binding sites. It has to attach to the membrane before it can exert its effect. So if those binding sites are blocked by the dirt, by any chemical, obviously the efficiency of disinfectant will drop significantly. So what does that imply? That implies that cleaning the surface, not disinfecting, don't mix them up, but cleaning the surface, just wiping it down, maybe using soap before you apply the disinfectant can be absolutely critical, depending on how badly the surface is contaminated with dirt, depending on the location, obviously the circumstances, but uh, don't fall into this misconception that just applying disinfectant will take care of everything. No, you have to clean it first to get rid of all those potential, uh, I guess, organic and inorganic molecules that can block the activity of disinfectant. And only then disinfectant is applied for its maximum activity, right? Um, as far as like this factor here, basically, well, what you need to understand is um, you have to know, let's put it this way, you have to know the microbiological profile of the surface or whatever the facility you work in. What does that mean? Depending on the facility, depending on what it does, depending on what kind of exposure to what kind of soil maybe, or I don't know, if it's we're talking pharmaceutical facility, what kind of personnel is getting exposed to the product. Each facility, each room may have a different microbial profile. So each room may have different organisms, or different facilities may have different organisms living in there on a regular basis. Remember how we discussed that, for example, air atmosphere can be heavily contaminated with spore formers, the bacteria that um, can be present in spores and survive in the spores in the air. So knowing the profile, what kind of bacteria to expect in a specific location, actually gives you a lot of clues as to how to treat that site. Because different bacteria obviously have different levels of resistance. They have different structure of their cell membrane, for example. They have different structure of their cell wall potentially, right? Or some may have cell wall, some may not have cell wall. So all of those factors obviously may impact, and quite often will impact, the susceptibility of bacteria to specific agents. So if you don't know what kind of bacteria you are dealing with in this particular setting, you have no idea what to use, right? Or you just will be using something generic or something basically working blind, right? But if you know, for example, that you are dealing with most commonly you're dealing with, say, biofilm former in a specific location, you probably will you should definitely account for that with your choice of disinfectant. Or maybe you will make a decision to scrub the surface. Because in this case, in this particular case, the biofilm formation can be a huge factor, right? Applying disinfectant by itself may not work if your bacteria is capable of forming biofilms. So basically, knowing what kind of pathogen, or not necessarily a pathogen, but what kind of bacteria you're dealing with, obviously gives you a lot of clues as to how to treat that bacteria. Because remember, all of them have different structure of the membrane of the cell wall, and so on and so forth. So not every disinfectant will be equally efficient. So blanket kind of application of disinfectant, sometimes it works, obviously, especially if you're using a more harsh disinfectants. But in some cases, it may not work, and the efficiency of disinfection will be compromised. So in certain facilities, for example, not necessarily related to pharmaceutical settings, I know for a fact that, say, nuclear plants, they have a team of microbiologists that all they do is they just profile their different facilities for microbial contamination, so that they know what kind of bacteria lives in there and what kind of problems that bacteria may cause to integrity of the systems, different systems, pipes, and whatnot, right? So not to mention the pharmaceutical settings where bacterial contamination uh, is of a paramount importance, right? Or avoiding that is so important. So knowing the profile, the most common contaminants, what kind of contaminants, what kind of level of resistance they have. Is it something significant, or is it just a standard kind of non-resistant species? The more information you have, the better. Um, Presence of different divalent metal ions we discussed earlier. So calcium, magnesium can serve what you need to know here. All these and some other ions can serve as cofactors for bacterial enzymes. So those cofactors mean that they are they are capable of making certain enzymes more efficient. And if those enzymes are involved in decomposition of disinfectants, obviously you don't want to be uh, contaminating your or giving the bacteria, exposing the bacteria to those ions. So that can all impact the efficiency of disinfection schedule. Alrighty, so that's that. So microbiological challenge, actually, keep forgetting it every time. This is what I was just explaining, knowing what kind of bacteria you're dealing with, how resistant it is it a biofilm former, or if it's a free cell suspension. All of those things matter. It also helps you choose the specific chemical. Different chemicals have different mode of action. Even the ones that rely on oxidation of the cell wall, they do it differently with different efficiency. So knowing what kind of bacteria you deal with may help you choose more harsh of a disinfectant or less harsh of a disinfectant, and so on. And then keeping the resistance profiles, keeping track of the resistance profiles of bacteria that are commonly present in your facility is also important. If it's a non-resistant species or poorly resistant species, it's one thing. But if you got some sort of a really resistant one, maybe it's time to use a more harsh disinfectant, right? So knowing the enemy, so to say, is exceptionally important. Um, in teaching labs, what we have in college, we obviously don't do any of that, but it's a different facility. Exposure times matter, obviously, depending on what you're dealing with. Concentrations are affected. Like I said earlier, if you are dealing with something more resistant, you want to be increasing the concentration of disinfectant to ensure maximum efficiency. So all those things. Gram negatives, gram positives, yes. So that all highly resistant species. that Mycobacterium tuberculosis is an example of, as I told you earlier. Gram negatives, make a note here, well, just as it says here, are usually more resistant than gram positives, with lots of exceptions, but it's not a, like I said, 100% thing, but definitely um, gram negatives are generally more resistant than gram positives. Why is that the case? Very easy to remember, because gram negatives have double layered cell membrane. Um, it's very tough for disinfectants and other antimicrobial agents to get through the double layered cell membrane, especially for antibiotics, because they actually need to get inside of the cell. Disinfectants don't necessarily have to. But like certain antiseptics, preservatives, antibiotics, getting through the gram-negative cell membrane, 2 layers of it, is very tough. It's very tough. So that adds to resistance, obviously, of the species. We're not going to talk much about the Mycobacterium tuberculosis. It's not important about as far as the intrinsic acquired resistance is concerned. Um, let's skip it for now. We will discuss this in a lot of details later in the course when we talk about the antibiotics. It's just this part here is as what I told you earlier. Mycobacterium tuberculosis is just an example of one of those species that is very famous for its high levels of resistance. It's not the only one. Don't by any stretch of imagination. Tons of E. coli that are very resistant. Acinetobacter baumannii is a very well-known pathogen for resistance. Pseudomonas is like one of the best because of the biofilm formation ability. So Mycobacterium tuberculosis is just the one of those famous ones. It's generally highly resistant species. Spores, as always, spores is always an issue. So we talked about it sufficiently. You want to be using intermediate or high level of disinfection in order to get rid of the spores, or maximize your chances rather to get rid of the spores. So this I guess flow chart or whatever you want to call it, must know for the test, gives you an approximate chart for the levels of resistance of different um, microbiological entities. Again, with lots of exceptions, but it's a trend. Know the trend. I'm not going to be asking for any. We're not going to be studying any. It's just too many of them. It's no point. But the general trend is this: bacterial spores are usually more resistant than fungal spores, and the fungal spores are usually more resistant than vegetative forms of fungi and bacteria. Okay, vegetative meaning the ones that are not spores, single cells communities. Within fungi and bacteria, you can make a note here with a lot of exceptions, obviously, but bacteria is usually a bit more resistant than fungi on average. So it would be bacterial spores, fungal spores, vegetative bacteria, vegetative fungi last. Okay, know it for the test. There will be definitely a question on that. As far as the viruses are concerned, very briefly, we're not going to concentrate on the viruses. Non-enveloped viruses have high levels of resistance, usually, again, with exceptions, as always, than enveloped viruses. Hepatitis A is one of the most resistant ones. So again, knowing the profile, knowing what kind of viruses you might potentially deal with at a specific location. Obviously, one thing it's HBV, it's not that resistant, or HIV, it's not that resistant. It's very easily removed. But hepatitis A is actually very resistant and requires much more harsh chemicals. As far as everything else about you don't need to know that. It's just the details we don't need them. Alrighty, intended application. Last, I guess, few points, last few slides as far as the intended application is concerned of the disinfectants. Long story short, what this is about, this part of the lecture is about, you must know what you are using your antimicrobial for, okay? Whether it's for the as a preservative, as an antiseptic, or as a disinfectant. We discussed it earlier. This will govern the concentration. This will impact the concentration of the chemical you're using. Preservation requires the lowest, antisepsis requires the second lowest, disinfection requires the highest. Uh, whether you are going to be exposing any biological tissues to your antimicrobial, we discussed that before. Concentration levels, depending on the application, depending on the um, level of disinfection that you're providing. So you need to know those values, not for the test I'm saying, but just in general when you work with disinfectants. Route of administration of the product of interest if you're talking about the preservatives. So if something is getting consumed, if something is getting applied to the surface of the skin and so on and so forth. Are you looking for inhibition of bacteria or just inhibition, or you need actually cytal activity? Reduction of concentration. So all of those factors obviously impact the choice. Certain chemicals, there's a lot of actually factors here. It's not everything here. Some disinfectants have different, for example, you can add it here, couple of points. Some disinfectants have different penetration capabilities through the organic and inorganic matter. For example, if you're trying to apply disinfectant on a contaminated surface, um, like contaminated by dirt, alcohols have very low penetration into organic dirt. So that disinfectant will be deactivated, or the efficiency of that disinfectant is going to be very low on a dirty surface. Um, while, for example, peroxides or aldehydes used as disinfectants have much better penetration into organic matter, and therefore you can sometimes even apply it to lightly contaminated surfaces without even cleaning the surfaces. So those things. Stability of disinfectant also matters in terms of temperature, in terms of pH sometimes. Um, You have to consider that certain disinfectants, especially in pharmaceutical settings, make a note of that. Some disinfectants can be very harsh in terms of their chemical nature, and they can actually affect the integrity of the surface that you apply it to. And in pharmaceutical settings, as we discussed a couple of weeks ago, it's a big factor. In other words, you don't want to compromise the integrity of your benches or anything else really in the pharmaceutical settings, because if you compromise the integrity of of the bench, it will inevitably lead to accumulation of contamination over time. That's why those surfaces are being made smooth, impervious, as we discussed, right? To minimize um, accumulation potential of microorganisms. So you don't want to be using disinfectants that will so to say, eat out your surface and create pores and crevices and whatnot. You may not see the negative effect immediately, but over time you definitely will. Some other chemicals, some other disinfectants, the more active ones, will cause metal corrosion. So you don't want that either, right? That's the same idea. This will cause accumulation of bacteria. And then all sorts of technicalities. You don't necessarily have to memorize that, but uh, basically the impact of disinfectant on the product. So it may cause corrosion and may cause may significantly change the clarity of for example, lenses that people use in their eyes, for example, and so on and so forth. So texture of the fabrics, synthetic polymers. So basically, uh, ask yourself when you're using these whether your disinfectant is going to somehow react with the surface that you apply to. So that consideration is also there, even if it doesn't completely destroy the surface. But if it compromises its integrity, it may lead to accumulation of bacteria. Is it critical in your case or is it not? If it is, then you probably should go with another chemical with a different choice. So those kind of things. And lastly, certain chemicals, certain disinfectants can be actually absorbed by the surfaces. They don't necessarily damage the surface, but they just get absorbed by the surfaces. And as they get absorbed by the surfaces, what happens? You reduce the exposure of your potential contaminants, bacteria, to that disinfectant because the surface absorbed half of it or something. So you reduce the effective concentration of disinfectant this way, and you don't want any of that. So the surface should not be absorbing disinfectant, okay? Because it may alter the integrity. That can happen as well. But even if it doesn't, it still reduces the effective concentration of disinfectant if it's getting absorbed. You don't want any of that. Alrighty, and that's pretty much it. Um, just skipping ahead to double check. This website here, you don't need to know anything for the test. This is just the very good website that has. It's kind of like it's a safety website where you can find all sorts of information about not just disinfectants, but all sorts of chemicals as far as their how to deal with them. Their MSDS sheets are there, can be found there. So um, all sorts of side effects, all sorts of properties of disinfectants and whatnot. So just a useful link for you to have. Um, so just add it like volatility, short versus long-term exposure, and so on and so forth. I'm not going to be asking you anything from this website. It's just, like I said, a useful link to have. But as far as let me just, I think that's it. I think this is not covered. Let's actually cover this slide because I have somehow I don't remember. I think there might be questions on that on the test. Yeah. Yeah, let's actually cover this one. Um, so this will be the last slide. How do people evaluate liquid disinfectants? So after this, I will also cover um, the phenol coefficient technique for next week because the phenol coefficient technique will be on the theoretical test. You need to understand that for the theoretical test. Don't forget, it will be the lab on it next week, but there will be also a test. There will be a couple of questions on the test for it. So phenol coefficient is just one of the tests that is used to evaluate the efficiency of disinfectant, how strong the disinfectant is. It's not the best test. It's not the most accurate test, but one of them. In general, these kind of tests are called suspension tests. The best tests for evaluating the efficiency of liquid disinfectants are called suspension tests. And what you do with those tests, you just basically estimate the percentage kill, the cytal power of the disinfectant, percentage kill. So you quantify how much bacteria is getting killed when exposed to that or this or that disinfectant. Phenol coefficient does not do it. Okay, phenol coefficient is a semi-quantitative test. It gives you a certain number once you finish it, once you complete it, but that number doesn't tell you anything about the percentage kill. Okay, the phenol coefficient. So it has a limited value. Suspension tests, the good ones, the ones that are the most accurate, the most informative, will give you the percentage kill so that you know how many bacteria percentage-wise will be killed, should be killed once you expose the surface contaminated by the species to that disinfectant. And what I wanted to show you here specifically for the test next week, because I think there might be a question on that there. How to convert rather percentage kill into this parameter here, log 10 reduction. The percentage kill, you know how to calculate it. You've done it 2 weeks ago before the break when you did the preservative challenge test. You divide percentage kill, percentage survival are connected. Percentage survival, you take the concentration of species that survived and you divide it by the concentration of species that you had at the start. In your case, it was like day 7 concentration divided by day 0 multiplied by 100%. Day percentage kill, you take the difference obviously, so how much got killed, that is day 0 minus day 7, all concentrations, both right? Day 0 concentration minus day 7 concentration divided by day 0 concentration. So whatever has been killed with respect to initial value. And that gives you the number in percentage. So you know how you hear it in the commercial, for example, that Lysol, the best disinfectant, as we all know, so to say, right, has a 99.999% killing power, and they show it on TV. They say that's the best disinfectant there is. Blah blah blah, right? So all disinfectants uh, have this kind of efficiency. Um, that's the standard. All disinfectants should have household disinfectants should have a percentage kill efficiency at 99.99%. Lysol is no different. Okay. Uh, it's just the way to commercialize things, but all disinfectants should have that efficiency. So that means 99.99% from the initial level of contamination, from the initial bioburden levels, should be killed. What I wanted to show you here is this log 10 reduction. Sometimes in certain literature, in certain applications, you will see a log 10 reduction number or log killing power, sometimes instead of percentage kill. So what I wanted to show you here for the sake of the test and just to your overall knowledge, how to convert between the two. Because if you've never been exposed to it, it might seem like something new. It's actually pretty straightforward. I'll show it to you in a Microsoft Word again. It is super, super basic. A little bit of calculations, but not the kind you think. Not Nothing to do with the dilution schedule or anything like that. Just the conversions. All right, so you should be able to see the word. Yeah. So percentage kill. 99.999%. So that means so many bacteria from the initial bioburden level are getting removed. You can right away calculate the percentage survival. That would be 100 minus 100% minus that number. That gives you percentage survival. So what is this log 10 reduction? And what was the other one? The log killing power. So let's take a simple example. You have a disinfectant that has a percentage kill of 99%. Okay. Just for the sake of simplicity. That means its percentage survival is 1%. Okay. So you had initially, let's say, initial percentage was of any bacteria, not any bacteria, but before you apply your disinfectant to the surface, whatever the level of contamination is, it is 100%, right? That's what you have at the start, 100% base value. And then you kill, for example, 99, and then 1% remains from whatever was the original value. So the original value is always 100%. Could be 1.0 multiplied by 10 to the power of 2 CFUs per centimeter squared, for example, or it could be 10 to the power of 5 CFUs per centimeter squared. It doesn't matter. We're dealing with percentage here. So you always start with 100%. That's your initial day 0, whatever concentration level of contamination. So what does this refer to? Look at these two values. If you have a certain surface that had a certain level of contamination that we take as 100% initial level of contamination, and if you reduced by applying the disinfectant, you reduce that concentration from 100 to 1, right? You killed 99, you're left with 1. So what did you do? You reduced the bacterial concentration 100 fold. from 100% to 1. 100-fold reduction. Okay? Everybody sees that, right? What is a 100-fold reduction if you were to use a log10 conversion? Log10 should be a subscript. Log10 of 100 is how much? It's 2, right? Because 10 into the power of 2 gives you 100. So why are we using 100? Because not because the initial concentration was 100, but because we reduced the concentration from 100 to 1. So it's a 100-fold change. 100 divided by 1 gives you a 100-fold change. And in order to calculate the log10, you just take the log10 value of this fold reduction that we got. Our fold reduction was 100. Log10 of 100 is going to be 2. So for my example, simpler example with a 99% kill, log10 reduction for this case is going to be equal to 2. Okay. Log killing power is exactly the same thing. You would write it down like this: 2 log killing power. You're reducing 100-fold. 2 log means 10 to the power of 2. That's 100-fold. When you have a disinfectant that kills 99.9, that means you are left with 0.001% of bacteria, right? Initial was 100. So what is a log, or what is a fold reduction in this case? In case with the best disinfectant on the planet, which is Lysol, right? 100 divided 100% was the initial level divided by 0.001%. That gives you how much? That gives you 3 decimals and 2 more decimals. 100,000 fold. Shouldn't be a gap, right? 100,000 fold if you were to convert to log10 reduction. How many zeros? 5 zeros. So it's a log10 reduction of 5. Because it's 5 zeros. 1, 2, 3, 4, 5. 10 to the power of 5 gives you 100,000 fold. And that's what we got. Why 100,000? Because we started with 100% initial level of contamination, whatever it is, whatever low, whatever high, doesn't matter. It's 100%. That's what we start with. And we killed 99.999. That means we're left with 0.001. And we reduced from 100 to 0.001. That is 100,000 fold. 100,000 fold is 10 into the power of 5. So instead of writing out 9999 and all that stuff, people just use this number, 5 log10 reduction, or 5 log killing power. So that would be the answer to this question. What is the log killing power of such a disinfectant? It's a 5 log killing power, and that is the conversion. So know that. Or I could give you like percentage survival and ask you, which is probably even easier because it's one step closer. What's the log killing power? Initial is always 100, and then we reduced it to this number. So you calculate the ratio, 100,000, 10 into the power of 1, 2, 3, 4, 5. That's your 5. 5 log killing power. Pretty straightforward. Okay. Make sense? I think it does, right? It's pretty basic math. 100 is, if you had 99.9, that would be a 1,000-fold reduction, right? 1,000-fold would be a log power of 3, log 10 of 3, because 10 to the power of 3 gives you 1,000. So that's how these values are being used. It's just it's not necessarily intuitive, right? When you see this, why is it 5? What does this have to do with 5? That's the math behind it. Alrighty, so that's that. And I think, yeah, that's the only thing that I want to talk about. Acceptable, yeah, let's, the last, I guess, point, let's talk about that. Know this for the test as well. For all disinfectants, commercially used disinfectants, acceptable levels of killing, so killing power, should be for bacteria at least 5 log killing power. And that's what 5 or 6 log killing power. That's just the requirements on disinfectants. Okay, so that's what Lysol tells you. Like I said, every commercially used disinfectant should have that. For viruses, log killing power should be 4, so it should reduce it 10,000 times. 10,000 times. 10 to the power of 4. Somebody's asking, is MPN part of the test? Yes, MPN is part of the test, obviously, because we covered it last week. So yes, you will need to understand those calculations. There will be a couple of questions on that. Alrighty, so last thing for today is going to be the explanation of the phenol coefficient. Right. So yeah, let's just talk about the phenol coefficient. So you'll get into the details of how the phenol coefficient is set up in the lab. What I will tell you is to how to analyze the results. One important thing, it's going to be a little bit confusing, maybe. The phenol coefficient lab, we will only perform week 2 of the lab. But the manual that is posted for you has both weeks, week 1 and week 2. You need to know both for the lab and for the lab exam and for the theory test. For an actual lab, you will only be performing week 2 for different reasons. We're not going to discuss those. We are not doing lab week 1 anymore. So just week 2. So what is the phenol coefficient test? Well, I will explain it to you once you set it up and you get the results, how to read those results. So first of all, like I said earlier, phenol coefficient test is the test that is used to estimate the efficiency of disinfectant. It is, like I said, not the best test out there. Rydell-Walker method is its original name. It has been modified multiple times, improved, and so on. So Rydell-Walker method is the same as phenol coefficient. The test is not the most accurate in terms of it's not the most quantitative. Let's put it this way. It's not the most quantitative. It gives you a number at the end, but that number doesn't tell you anything about the killing power. What it does instead, that number that you get, and the number could be like 4, 5, 6 without any units. That number tells you one thing, how much more that disinfectant that you are testing is more efficient compared to phenol. So the phenol coefficient, let's say of 4, means that your test disinfectant is 4 times more efficient than phenol. Why phenol? Because phenol, why are we using phenol as a baseline? Because phenol was traditionally the first and the most widely employed disinfectant back in the day. And for the longest time, people were comparing the efficiency of new disinfectants to phenol as the baseline. So again, when you get this number 4, 5, 6 for each, like say citrus may have a phenol coefficient of 5. What does that mean? That means that your test disinfectant, let's say citrus, is 5 times more efficient than phenol. If some other disinfectant is 10 times more efficient than phenol, so obviously it is more efficient than citrus as well. So it's a relative scale like that compared to phenol. As far as the setup and reading the results, so when we look at this table, that's the only thing we're going to look at to not overwhelm you with the details. We will be looking only at the results in these 2 columns, 5 minutes and 10 minutes. And I will get to those in a second. The 2.5 minutes and 7.5 minutes, forget about them. We're not going to discuss them. So how is the setup performed, first of all, so you understand what's going on. And what I'm about to explain to you right now, this part will not be performed in the lab. Okay? This was usually the week 1 part of the lab back in the day when we actually ran week 1. We're not doing it anymore. You're going to get the results as if you ran the lab, but you actually didn't the first week. But anyway, how is that week 1 set up? What you do is you take your test disinfectant, let's say citrus, and you take your phenol as a baseline, actual phenol, and you create a bunch of dilutions of both phenol and your test disinfectant as per this table here, but these levels of dilutions are not written in stone. They can be different. But let's say we got these values. We decided to perform dilutions, these kind of dilutions. So you take your test disinfectant, say citrus, perform 1 in 400 dilution, 1 in 500 dilution, 1 in 600 dilution, 1 in 700 dilution, 1 in 800 dilution. You prepare them in the separate test tubes. And then you prepare these dilutions, as you can see, different levels of dilutions from these for phenol, okay, in another set of separate test tubes. Then what you do is you take a penny cylinder. You must have seen, a lot of you must have seen that penny cylinder before in the lab. It's basically a stainless steel hollow cylinder, small, fairly small, like 1 centimeter tall. You expose it to bacteria, whatever your test bacteria is, and you put that penny cylinder one piece at a time, one penny cylinder at a time, into your test disinfectant test tube with all of these levels of dilution. So you take one for 1 in 100 dilution, one penny cylinder exposed to bacteria, expose it to 1 in 500 dilution, 600 dilution, 700 dilution, 800 dilution, and so on. So one penny cylinder each. So in other words, what you're doing is you're exposing a surface, penny cylinder in our case, to a bacteria, and then you are throwing that surface into the disinfectant, different dilutions of that disinfectant. And then as soon as you put that penny cylinder into disinfectant, you start the timer like a clock. So you time it carefully, the exposure of your bacteria to that test disinfectant. And what you do next is after 5 minutes, you usually have separate sets of test tubes, like several sets of test tubes. After 5 minutes, you transfer your penny cylinder from 1 in 400 dilution into recovery media, just simple media, nutritious media, to allow bacteria, any bacteria that survived, to recover. And then you also have a second set of test tube that has 10 minutes, that has been where the penny cylinder would be exposed to test disinfectant for 10 minutes. And then after 10 minutes, you transfer your penny cylinder into the recovery media that doesn't have any disinfectant, just the recovery media. And then you do the same for all of these dilutions for test disinfectant and for phenol as well. What's the point of this? We want to basically know if we basically want to answer a simple question. If we expose our bacteria to this level of dilution of disinfectant for 5 minutes, are we going to be able to kill all bacteria that were present on that penny cylinder or not? And are we going to kill all that bacteria after 10 minutes exposure when we increase the exposure time? And then we do the same. We ask the same question for different levels of dilution, 1 in 500, 600, 700, 800. So as you can see, you progressively increase the level of dilution. So the efficiency of disinfectant drops as the level of dilution increases. And then you just expose penny cylinder to it, and then you move it over to recovery media. And the idea is, if disinfectant at this level of dilution was efficient enough to kill all bacterial cells, then in the recovery media, you're not going to be able to see any growth. The recovery media, which will get incubated like overnight or like for 11 hours, 8, 9 hours, you look at the media. If all bacteria was killed, there will be no growth in there. Test tube will be completely see-through. If, on the other hand, for this particular dilution or any other dilution, this level of dilution was not sufficient to kill bacteria completely. In the recovery media, bacteria will start replicating. It will heal its wounds, so to say, right? And it will replicate. And as a result of that replication, that test tube will give you growth, will give you turbidity. Okay? After you incubate these test tubes overnight, you take them out of the incubator and you look at growth in the test tubes. And you make this kind of a table where NG or minus sign stands for no growth. So that means completely see-through test tube. And G or plus stands for growth, positive growth. So again, if you take out the test tube and there is no growth, that means this level of dilution at 5 minutes exposure time was able to kill every single cell in that test tube on that penicillin. And as a result, we don't see any growth after overnight incubation. Okay, so you collect all this data after 5 minutes exposure of your bacteria to test disinfectant and after 10 minutes exposure. And you write down the results. And you get something like this, obviously hypothetical. Just an example, pretty ideal data, but that's what you might get once you perform week 1, which you will not be doing, but I'm still explaining it to you. You still need to know it. So how do you analyze these results now? The question is. You basically look at this pattern of negatives and positives. Again, negative means bacteria was killed. Positive, it means some bacteria survived. We don't care how much survived, as long as at least some survived. They will give you growth, and we will register that growth, and we will put it in the table. We don't care how many survived. We're not quantifying that. So the same applies obviously for the phenol. Exactly the same thing, just a different chemical and a different dilution, different dilutions. I will comment at the end why the dilutions are different so that you understand. So how do we analyze the results? Remember, we're looking only at a 5 minutes exposure and a 10 minutes exposure. So what you're doing now, you look at this data, well, I guess before you analyze it, one more thing I wanted to mention. When the results work out properly for this test, you should be getting something like this kind of a pattern. So what kind of pattern am I talking about? For the highest concentration of disinfectant, for the lowest level of dilution, 1 in 400, right? This is the highest concentration of test disinfectant out of all these 5. 1 in 800 obviously is more dilute than 1 in 400. So this usually the top concentration is still sufficient, is still efficient enough, and it will kill all bacteria that was exposed to test disinfectant, both at 5 minutes and 10 minutes. As you go down in this column, as you can see, the level of dilution increases, which means that the level of the concentration of test disinfectant decreases, right? 1 in 400 is higher than 1 in 800 concentration, right? So as you go down in this column, in other words, as you decrease the concentration of test disinfectant, the efficiency of that test disinfectant obviously drops, right? Because the concentration drops. So that means in case of an ideal results, as you go through your results, you should be seeing something like this kind of a pattern. Full elimination of bacteria for the first few dilutions, maybe 1, maybe 2, maybe 3, but you should have negatives on top, eventually transitioning into positives. Why are they transitioning to positives? Because as the concentration of disinfectant drops, its efficiency drops, and at some point, 5 minutes exposure is not going to be sufficient for the disinfectant to kill all the cells that you expose to that disinfectant. So that's why negatives, see-through test tubes transition into actually test tubes with growth, meaning that some survived and therefore gave you growth. For the 10 minutes exposure, the pattern is exactly the same, except for you should probably see more negative signs than you see in the 5 minute test tubes. Why? Because the exposure time is longer. So obviously, as we discussed earlier, the longer the exposure time, the more efficient the disinfectant will be. So with a 10 minutes exposure, disinfectant will be able to still kill bacteria when you expose it, even at lower concentrations. So as you can see, for 1 in 600, 5-minute exposure is already not sufficient to kill all bacteria. There's some growth already in the test tube. But for a 10-minute exposure, concentration is the same, but the bacteria is all killed. Why? Because we exposed it for longer. Disinfectant had more time to deal with bacteria. So that's what ideal kind of set of result looks like. Exactly the same logic for phenol. No growth on the negatives at the start, and then eventually transitions into positive. And the same deal, your 10-minute exposure will have more negatives than positives because it's a longer exposure. As far as the analysis goes, so how do you analyze it? In order to analyze this, you are looking for a so-called critical dilutions. Critical dilutions for test disinfectant, and it's going to be one value. And you're looking for one critical dilution of a phenol for the phenol. So what is a critical dilution? Critical dilution is the highest concentration of the highest dilution, the highest dilution of the test disinfectant that as well as phenol that kills bacteria at 10 minutes but doesn't kill at 5. Okay, so kills at 10, doesn't kill at 5. Same applies for phenol. The highest dilution that kills at 10 and doesn't kill at 5. So let's go with test disinfectant. Highest dilution, so that means we're going to be looking at the bottom. We'll start looking from the bottom because that's where the highest dilutions are. The highest dilution that kills at 10 doesn't kill at 5. So, okay, 1 in 800. Does it kill at 10 and doesn't kill at 5? No. So this one is out of consideration. Next one we're looking at kills at 10, all negative, doesn't kill at 5. Awesome, that fits our criteria. Next one kills at 10, doesn't kill at 5. Fits the criteria. The last two don't fit the criteria because they don't kill. They kill at both 5 minutes and 10 minutes. So we are left with two values: 1 in 600 dilution and 1 in 700 dilution. Kills at 10, doesn't kill at 5. Now the question is, which one is the critical dilution? As per definition, the highest dilution. Out of these two, 1 in 600 and 1 in 700, which one is the highest? 1 in 700. So that means for this set of results, the critical dilution for the test disinfectant is 1 in 700. So what you do is you write this value down and you claim critical dilution of test disinfectant is 1 in 700. For phenol, you do exactly the same thing. Exactly the same thing. You look for the critical dilution. So let's look at phenol. Kills at 10, doesn't kill at 5. Last one doesn't apply. The next three apply. This rule applies, right? Kills at 10, doesn't kill at 5. Kills at 10, doesn't kill at 5. Which one is the highest of the three? Obviously 1 in 110. That's higher dilution than 1 in 05, 1 in 100. So critical dilution of phenol for this set of data is 1 over 110. Okay? And the very last step, the final calculation, super simple. How do you calculate the phenol coefficient? You take the critical dilution of phenol that you just found, and you divide it by the critical dilution of test disinfectant. So in our case, we will be dividing. I can show it to you. Oh, it's okay. Let me reshare the screen. I'll just write it down on the somewhere here. To find some room. Okay, I'll just add it here. So phenol coefficient in this case will be, like I said, critical dilution of phenol divided by critical dilution of test disinfectant. the ratio flips. You end up getting 700 divided by, sorry, not 100, not 100, obviously. That's not what I meant, 110. This one, 110, divided by 110. And that will give you 6 point, I don't know, 8 or something like that. Maybe 6.7, something like that. You can calculate it more accurately. Right? No, it's actually less than that, 660, something like 5, I guess, close to 5. Even 4, maybe. You can do the math. Okay, something like that, 6.4, yeah. What is it? 6.36, okay, so pretty close. So that's going to be the phenol coefficient based on this on the data that I gave you in this example. What is it? What does it mean again? The only thing it means that test disinfectant citrus is 6.36 times more efficient than phenol. That's it. Killing power, all that jazz. We don't know what that this test doesn't give you those values. All it gives to you is this comparison. Okay. So again, for the lab, you're not going to be setting this table up and getting these results. We used to do that, but not anymore. You'll be doing a second week of this lab. You know what I'm going to do is I probably will record like a separate 10 minutes video explaining the second week just in case that people have it as a reference. Second week setup is very simple, even simpler than this and faster than this, but I'll just provide some background information so people understand what exactly they're doing. As far as the test is concerned, the theoretical test next week is concerned, what do you need to know? You need to know both week one. Just let's do just week one. You need to know how to calculate phenol coefficient. Let's stick with that. How to calculate phenol coefficient based on this kind of a data. 5 minutes and a 10 minutes exposure data. 2.5 and 7.5, forget about them. They're not that much different. It's just people test disinfectants at different times. All there is to it. We're usually doing 5 and 10 minutes. Okay, so you will need to be able to calculate phenol coefficient based on this kind of a data, and I just explained how to do it. Don't forget what is a critical dilution. It's also in the lab manual. You can find it and how to find that critical dilution. Highest dilution that kills at 10 doesn't kill at 5. If you have several candidates for it, you take the highest dilution as per the definition, and that would be 700 in this case, and 1 in 10, or 110, yeah, 1 in 110 for the phenol. And then you divide one by the other and you get the number. Phenol coefficient should always be greater than 1. So if you make a mistake and you divide 110 by 700, it's going to be a value that is less than 1. As soon as you see that, you know you made a mistake in plugging in how you plugged in the values. It should be the value that is greater than 1. Because modern test disinfectants are much more efficient than phenol, and they're a lot more efficient. Chemicals that are being used are a lot more potent, and so on. And that is the reason why these dilutions here for the test disinfectant are higher than the ones for the phenol, because test disinfectant is more efficient than phenol. So we need to dilute it even more. If we diluted test disinfectant to the same levels as phenol, all of these values would have been negative. Okay, because test disinfectants are usually much more. The modern day disinfectants are a lot more efficient than phenol. Phenol was used for the longest time traditionally, and this whole methodology is comparing test disinfectants to phenol because of that. Okay, so that's pretty much the whole method. You'll be given a table on the test next week. That is theory test and midterm too, and similar table, and you will have to tell me what the phenol coefficient is. That's it. All right. Alrighty, guys. So that's going to be it a little bit early today, which is good. So 20 minutes early. But that's always how much we cover for the second midterm. So again, next week, Friday, 7:30. I'll post it on on Luminate. platform, pencil, calculator, be on time. It's going to be the same setup, the same 3 sections in the other room, everybody else in 203. Be on time, I guess. Yeah. So everything we covered so far after midterm 1, obviously. Midterm information from midterm 1 will not be on the test. The tests are not cumulative, so you only have 2.5 lectures to study, if I'm not mistaken. Environmental monitoring, water, and whatever we did today. Everything we did today, and the calculations. 10 calculation questions, as always. Calculation questions are very, very similar to the ones we had in the first midterm. So if you got those ones, or if you at least know where you made the mistakes, you should be able to do this much, much better in the second midterm. Study in advance. There's only 1 week remaining. Hopefully people already started studying. I mean, maybe not, but a lot of people got not so good grades on the first midterm, so you must improve for the second one. Because then after that, it's only 1 midterm remaining, which, I mean, final exam remaining. So take it seriously, the second midterm. Now that you know what the questions are like, what the calculations are like I told you at the start of the semester, the second midterm always has much higher, well, I don't know, much higher, but higher average than the first one. Hopefully it's much higher because, yeah, a lot of people need to improve for sure. Kyle, no. The Schick-Martin test is not Schick-Martin test is not included. No, only what I spoke about today. Everything else we will cover after the break, after the midterm. Okay. Alrighty, guys. So that's going to be it. If you have any questions while you're studying, while you're preparing, you can always email me. But yeah, that's about it. Okay. All right, guys. So have a good weekend and start studying for the test. And I'll see you next week in class for the test. The test is in person as always. So see you in college next week. See you.